So chapter 17 is going to take what you know about cell division, about what you know about DNA and chromosomes and all of that, and is going to give some real things that you can actually see. So we're going to talk about heredity and how what we know about DNA influences what characteristics are getting passed on. So the first thing that I thought that we would talk about is the idea of selective breeding. Selective breeding has been happening for thousands of years. So this is not a new fangled technology that is based on some discovery that we've made about DNA. Selective breeding has been happening for as long as humans have been breeding animals, basically. And it could be animals or it could be plants. But selective breeding is the process of breeding organisms, animals or plants, based on, and it can be based on two different types of things. It could be based on their qualities or their behaviors. So selective breeding, which is very important for a lot of things that happen near us, lots of people around this part of the country breed animals, whether it's horses or cattle or some other type of animal that lives on a farm or a ranch type setting. It could be for the food industry, so there could be agricultural things there. But this idea of selective breeding has been going on for a long time, and I'm sure will continue to occur because there are organisms that have desirable qualities that we would like to use or exploit. So I'm showing an example here of a wolf. So this is Canis lupus lupus. It's a wolf. And a wolf is in the same family as these three things, which are all Canis familiaris. And so selective breeding, a really good example of it is dogs. There are so many types of dogs. Based on what we know now about DNA, it appears as though all dogs at some point were descendant from a wolf or a wolf-like organism, that was their ancestor, and that based on reproduction and breeding, different races of dogs have been produced. Now, just as a side note here, do you all know what kinds of dogs these are? Maybe we'll have a little doggy lesson here. So the first one is like a Great Pyrenees or just a Pyrenees if you want. The second one is my dog. It's a miniature dachshund. She's adorable. And the third is a toy poodle. And they're all bred based on very different reasoning. The first one is sort of bred as like a protector. So this is a really good dog to have if you have a lot of land and you want to keep the animals on your land safe, or you want to keep them protected from predators. My dog, if you want a dog that does not dig holes, don't get this dog because she digs holes. Dachshunds are well known for their digging ability, and they used to be used to hunt badgers, like things that live in holes under the ground, because they're really good at digging, and they're long and skinny, so they can fit in. And toy poodles, well, gosh, they're just adorable. That would probably be one of the reasons that they were bred that way. So for some of them, it's their behavior. For some of them, it's their appearance. But selective breeding has been going on forever. And now that we know a little bit more about how heredity works, selective breeding can get a whole lot easier. So there are some people that have had some ideas in the past. In the past, scientists and people have thought that, first of all, somehow the whole organism just had its characteristics and they didn't come from anywhere and evolution wasn't a thing. Then some scientists thought that it was only the sperm cell that determined what characteristics the offspring would have. So this is Van Leeuwenhoek. Maybe you heard about him in Science 10. He did a lot of microscope-related stuff. Then you have Regnier de Graaf. He thought that it was the egg cell that was determining the characteristics of the offspring. But in 1860 or thereabouts, a fellow named Gregor Mendel, and he's important, that's why his name got written in capitals. In 1860, Gregor Mendel did a bunch of experiments, and he determined that the two parents both contributed to the offspring characteristics. So up until that point, we weren't totally sure what was determining whether or not, say, the dog that you got from breeding two dogs was going to look like the mother or the father. Because in some cases, it would seem like, oh, the dog looks exactly like the mother. It must be the mother that determines the characteristics. Whereas in other cases, we would say, oh, well, this one looks just like the dad, so the dad's characteristics must be the ones getting passed on. And I'm sure if you think about in your own family, maybe not you personally, but there's probably someone in your family where you're like, oh yeah, that person totally looks like their mom and doesn't really look like their dad at all. Or that person totally looks like their dad, doesn't look like their mom at all. And so because if you just look at the outside, sometimes it's hard to tell where the characteristic is coming from. It took until we understood a little bit more about genetics for us to really understand how to make the most of selective breeding. So Mendel's name is the one that is the most important. We'll often use Mendel as an adjective. So we'll use the word Mendelian as an adjective to describe this type of genetics. So he's a pretty famous guy. His name will definitely show up in a question. We will talk about the experiments that he did. And so it would be legitimate and perfectly fair for me or the diploma exam to ask you specifically about Mendel and what he did, because his experiment is sort of the basis of this whole entire section that we're doing. So what did he do? The picture that you see here can be found on page 588 of your textbook. So there's no need to recopy it. I just wanted to have it here so I could refer to it. The picture in your textbook isn't identical to this, but it has the same information. In Mendel's experiments, he always started with a pure cross. So for Mendel to do experiments about genetics, he had to make sure that the plants that he was starting with were pure. That means they had all the same copies of genes. They didn't have different versions. So before any experiment, he did this. He let the pea plants, which is what he's using, auto-fertilize. Now in plants, it's not so weird. Go ahead, fertilize yourself. That means your DNA fertilized, or your, if you're the plant, your sperm cell fertilized your egg, so the DNA has to be the same. But he let the pea plants auto-fertilize so he would be sure that they were genetically pure. And so before he did any experiment, he always let the plants auto-fertilize themselves to make sure that the DNA strain was pure. The P generation stands for parent. And that's what he did with those auto-fertilized pea plants. So the P generation were the parents, and they were always genetically pure. That meant that they would always have 2 copies of the same gene. So he always made sure that that's how his experiments started. Then he chose 7 traits. That kind of looks ugly. Let's do that again. 7 traits. So the 7 traits that he chose were flower color, flower position, seed color, seed shape, pod color, and stem length. So he chose 7 traits that he could see 2 different versions of. And with those 7 traits, he would cross opposite parents. So he would find 2 of these parent generation plants where they had just auto-fertilized themselves. And he would find plants that had the opposite characteristic. So for example, he would take a purple and a white flower, opposite characteristic. Or he would take 2 plants that had axial versus terminal flowers. If you take a look, terminal means the flowers are at the end of the stem, and axial means that they're somewhere lower down in the middle part of the stem. But he chose 7 traits, and he would cross parents that were opposite. The F1 generation are the descendants of the parent generation. So after we crossed 2 parents that were opposite, we get the F1 generation. And this is pretty typical, typical way of naming things. So regardless of whether we're talking of Mendel or of some other experiment, P would always be the parent, F1 would always be their descendants, and then we would just increase the number, F2, F3, F4, depending on how far we were going to go. The F1 generation was the result of a monohybrid cross. A monohybrid cross, like the word suggests, is a cross based on one trait. Now we'll see later on that we can, of course, breed organisms based on more than one trait, but we will start by talking about monohybrid crosses, where we pick one characteristic that's different between the 2 organisms and then breed based on that. Does anyone have any questions that they want to ask here? So what did Mendel discover? He discovered something pretty important about dominant and recessive genes. The F1 plants, all of them, so all of the F1 plants showed only one of the parental traits. So we're saying that if he crossed a purple flower and a white flower, all of the flowers were the same color. And he noticed that for all of the 7 traits that he had chosen. So for example, we're going to take the shape of the P. So I'm just going to go back here for 1 second. We're taking seed shape here. The seeds could be round or the seeds could be wrinkled. Those were the 2 shapes of them. So if we use that as our example, he crossed round seeds with wrinkled seeds. Those were the 2 parents. So one of the parents had round seeds, the other parent had wrinkled seeds, and he found that in F1 they all had round seeds. Which is weird. You would think that some of them would end up showing the characteristics of one of the parents. Because of this now, we have an operational definition of what the word dominant and recessive means. Dominant means the trait expressed in F1.